Hi guys, we're going to be talking about libraries today. So this is going to be kind of a briefer tutorial, and I just want to give you a rundown on what is a library, what are they used for. And we're going to start with an example using GFP. So if you can imagine that you have, the pen's not working, there we go. We have a construct that is promoter, GFP, and we're just going to think about only that. And let's just say this doesn't really give you that much fluorescence, that this promoter is kind of weak. Let's say it's a wild type and we put this in our cells and we don't see any color. We can use a library in order to find a brighter version of this. So what that would look like is we're going to have the same kind of overall shapes. We have our promoter and we have a GFP. So same thing. And what we might do, there's a lot of ways to approach this, but what we might do is make an RBS library. So RBS, ribosome binding site, would show up like right there. So we'd want to randomize probably a little bit more than this, but just upstream of the first codon, that ATG. Again, this is just conceptual. So if we randomize this RBS, we're going to be able to kind of modulate the amount of GFP protein that eventually gets translated. So if we target this area for mutagenesis, we're not going to go into how we actually do it, but let's just say that we can kind of wish into existence these base pairs right here. What we're actually going to have is a bunch of constructs that again have the same general format. So if I'm going to draw a line here, we have a promoter. And then I'm going to break my line, and we have GFP. And then instead of our wild type, let's say our wild type was, let's say CGGG. Instead of having that wild type, what we're going to see instead is a bunch of different versions. So we'd see something like this. Oh, that's way too big. Let's change that to, that'll work, whatever. Oh God, my thing's frozen. Okay, well, we'll just go with that. So we're going to have a bunch of different RBSs. So again, we've changed from a CGGG to a version that has AAA, TTT, ATAT, ATCG. So all of these are possibilities from these ends. So this is theoretically what we have with those ends. And then if we actually, like we said, if we just wish these sequences into existence, what we're going to have is a test tube, beautifully represented here. Looks more like a bag. But we have a test tube with a bunch of plasmids in it. And so if we just draw some plasmids, we'll say that each one of these plasmids is one of these. So if this is construct 1, maybe this plasmid is plasmid 1. This is construct 2, this is plasmid 2, construct 3, plasmid 3. So this plasmid has not the wild type like we saw up here, but it has this AAAA instead of the normal wild type RBS. This plasmid here, instead of having that CGGG, has this TTTT. So we all have, all these plasmids share the same basic shape and are described by this NNNN, but they all differ at those ends. All those ends actually turn out to be some sort of real sequence like AAA or TTTTT. So this is what a library is. It's this like one tube, a bunch of different versions of the same basic thing, or at least that's one definition. There's other definitions, but in this kind of tutorial, you have the same basic overall shape with a few different tweaks on it. And in this case, we have an RBS library. So all these RBSs are different. Again, so plasmid 1 over here has RBS 1, plasmid 2 has RBS 2, which is TTTT, and so on and so forth. Now, when you have that library, you're not going to know what's what. For all you know, these NNNNs could turn out to all be A's. So you could have all AAAA and TTTT, and you could be missing out on all the other diversity. That's unlikely if you get a high quality oligo synthesis, but it's possible if you're doing your own oligos and you mess something up, you may not have any diversity in that library. So right now we're at the point where we have a tube and it's full of plasmids. Each plasmid is unique, hopefully unique. Well, if we're randomizing only 4 base pairs, then we're going to have a lot of overlap. But if we were randomizing like 100 base pairs, then every plasmid would be unique. So we end up with this tube. We have our different plasmids in it, and now we want to figure out which one of these plasmids we want. And originally we said that we wanted to have a brighter GFP. That means we want to have a stronger RBS, something that's giving us more expression. So what we would do is we'd transform our little tube of DNA into cells. So I'll draw a few cells right here. So there's a cell, there's a cell. My cells look very similar to plasmids. Hopefully you don't get confused. And we'll switch to red. So plasmids are now red, cells are blue. So now we have each one of these cells has a plasmid in it, and maybe this cell gets plasmid 1, this cell gets plasmid 2, this cell gets plasmid 3, and we didn't make a plasmid 4, but we'll just say that cell gets plasmid 4. And then we take these cells and we spread them onto a plate. So now we have a big petri dish, and we have a few colonies, 1, 2, 3, 4, we'll say 5 colonies. Realistically, you're going to have way more than that. And then we shine a little bit of UVA light on them. This is our screen. We're screening for high fluorescence, and hopefully what we'll see is one of these colonies is going to be really bright. So let's say if we shine our UV light on it, this colony gets really, really bright, and it just pops out, and it's very easy to see. And these other ones, they're a little bit dimmer. So we don't like those dim ones as much. So we're not going to really pay much attention to them. We're going to kind of cross them out. But this one is really cool. So then what we do is, after we've screened, we'd go and we'd try to characterize it. So in this case, characterization is pretty simple. We just sequence it. And for argument's sake, we'll just say that it sequences to, let's see, how about TTTT? So TTTT. And if we go and we look at our little dummy theoretical sequence, we're going to find that TTTT is construct number 2, which is right here. So now we know what RBS mutant led to increased fluorescence. So that's one way you can use an RBS library in order to generate brighter cells and find a, I guess, altered biological activity. So hopefully that makes sense. Let me know if there are any questions or uncertainties, and I will try to answer them.